The lab is dim, and everything that matters is humming softly, like it is trying not to wake you. Air conditioner, incubator, a tired little fan, all keeping time with your slow breath. You might think the night shift is empty and nothing is really happening, but in a single drop of water, whole civilizations are working quietly, without ambition, without applause. Bacteria do not sleep. They simply continue solving problems the way a stone solves gravity. And one of their strangest solutions is almost tender. They keep tiny genetic keepsakes of past viral attacks, little clipped phrases stored in their own code. Tonight, with the Sleepless Scientist keeping the lights low, you will follow the patient trail of clues that led humans to notice this. Not quickly, not dramatically, but one calm, stubborn observation at a time. You are standing in a lab that smells faintly like alcohol, wipes, and warm plastic, and you are looking at DNA the way you look at a very long receipt, not because it is exciting, but because it is what is there. The first people who noticed CRISPR were not hunting for a miracle tool. They were doing the quiet work of reading genomes, line by line, base by base. They were trying to understand what makes a bacterium, and what makes one strain different from another. Then they found something that did not behave like the rest of the code. It was a pattern that looked too neat to be an accident, a short sequence, then another copy of the same short sequence, then another, and between these repeats, little odd gaps, spacers, unique bits, like someone had pasted tiny clippings between identical strips of tape. You can picture it as a child making a scrapbook. Every page has the same decorative border, but the photos in the middle keep changing. Back then, that was the whole mystery. Not what it did, just why it was there. Because bacteria, despite their reputation, do not waste space for fun. Their genomes are usually tight, efficient, less poetry, more spreadsheet. So when you see a region of repeated DNA with variable spacers, you squint, you mark the page, you write a careful note in the margin, and then you move on, because you still have millions of letters to read. This is how big discoveries often begin. Not with thunder, with a scientist muttering, "That is odd," and then going home for dinner. The repeats were noticed in the late 1980s in bacteria like Escherichia coli. At first, they were just strange repeats, a curiosity, the kind of thing that gets a small paragraph if it gets anything at all. Over time, more genomes were sequenced, different bacteria, different archaea, those tough, ancient microbes that treat boiling acid like a spa day. And again, the repeats. Repeat, repeat, repeat. Spacer, 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 spacer. Same architecture, different middle pieces. It was consistent enough to feel intentional, but nobody knew what the intention was. And the universe, as usual, was not eager to explain itself. So, you watch the pattern show up across species. You watch it vary in length. You watch some bacteria have long arrays with many spacers, and some have short ones, like they have not been keeping records for very long. And you start to wonder, are these spacers random? Or are they collected? This is where the story begins to feel slightly personal, because you, lying there in the dark, also keep spacers. Not in your DNA, no. In your mind. Little clipped moments, a sharp sentence you still remember, a mistake that still has teeth, a warning stored somewhere deep that you do not fully examine until it is needed. Bacteria do not have minds, but they do have problems. And one of the biggest problems in their world is viral predation. If you want to understand those spacers, you need to meet the things that force bacteria to remember. And unfortunately, they are everywhere. In a drop of seawater, there are viruses by the millions. Not the kind that make you sneeze, the kind that hunt bacteria. Bacteriophages. Phages, for short. They are small, hard, and strangely elegant. A protein shell, like a tiny capsule. Sometimes, with a tail, like a lunar lander. They drift until they collide with a bacterium. Then, they latch on. Not lovingly, they puncture the cell wall. And they inject genetic instructions like a theft. A bacterium is not built for drama. It is built for metabolism, for turning sugar into motion, for dividing, for surviving the next minute. So when a phage arrives, it is not a philosophical event. It is an emergency. The phage genome enters, and the cell's machinery is hijacked. Ribosomes start translating viral proteins. The cell becomes a factory that does not get paid. New phage parts assemble inside the bacterium, quietly at first. Then the end comes fast. The bacterial cell bursts. Lysis, a clean word for a wet outcome. And the new phages spill out to hunt again. This is not rare. This is the background weather of microbial life. A constant pressure, like wind on a coastline, shaping everything without asking permission. So if bacteria have a way to defend themselves, it will matter. Not symbolically, not someday, immediately. But for a long time, the repeats did not look like defense. They looked like nonsense, or maybe regulation, or maybe a leftover from ancient evolution. Scientists tried to guess. They were wrong in several creative ways, which is normal. Nature does not label its parts. It just ships the product and lets you suffer. Eventually, someone noticed something subtle. These repeat and spacer arrays were often near certain genes. Genes that looked like they encoded enzymes, proteins with motifs that suggested cutting, unwinding, handling nucleic acids. The cell's paperwork department. These genes would later be called Cas genes. CRISPR-associated genes. Cas, a short name for proteins that do very sharp work. But even then, the big question stayed gentle and stubborn. What are the spacers? Where do they come from? And why do they change? Imagine you are a geneticist in the 90s. You do not have the smooth, automated sequencing pipelines of today. You have gels, bands, careful pipetting, a life spent trying not to contaminate tiny tubes with your own skin cells. You sequence a CRISPR region from one strain, then from another. You align them. The repeats are similar. The spacers differ, and sometimes strains that are closely related share some spacers, like they have a shared history. But then, one strain has extra spacers at one end, new ones, as if the scrapbook keeps growing, always adding at the same side. That detail matters because it implies direction, not random chaos. A process, a mechanism. And mechanisms in biology usually exist because they help you not die. The term CRISPR itself arrives in the early 2000s, clustered regularly interspaced short palindromic repeats, a name that feels like a committee tried to win a bet, but it describes the structure. Clustered meaning grouped together. Regularly interspaced, meaning repeats separated by spacers like beads and knots. Short, because they are not huge sequences. Palindromic repeats, meaning the repeats read similarly forward and backward in a way that forms shapes in RNA later. Even the name is quiet and descriptive. No poetry, just a label pinned to a mystery. And now, because the pattern has a name, it becomes easier to talk about. It becomes easier to collect. And slowly, the scientific world starts doing what it always does. It starts building a file folder labeled Weird but Consistent. Then comes the moment when someone does the most dangerous thing in science. They compare. Not emotionally, computationally. They take spacer sequences and search databases. They ask, has this little snippet been seen anywhere else? And at first, nothing. Because databases were smaller then. Because the microbial world was still mostly unsequenced darkness. But as more viral genomes are added, as more plasmids are cataloged, as more genetic debris is collected, some spacers begin to match. A spacer from a bacterium matches part of a phage genome. Not perfectly always, but unmistakably. Like hearing a phrase you recognize from an old argument. Like finding a sentence copied out of a threatening letter. It is one of those quiet moments where the room does not change, but your understanding does. Because if the spacer matches a virus, then it might be a memory. And if it is a memory, then the whole CRISPR array might be a record of infections. A list of enemies, not carved into stone, written into DNA. The bacteria in this picture are not passive victims. They are archivists. They keep samples. They save fragments. They file them away in a repeating framework that can be read later. And yes, it is almost tender in the way a trap is tender, in the way a locked door is tender, in the way a scar is tender, because it is evidence that something hurt you once and you adapted. Before we get too sentimental, remember what this means in practice. A bacterium has been invaded by a virus. The bacterium survived somehow. And during that survival, it captured a piece of the invader's genetic code and inserted it into its own genome. That is not a metaphor. That is a physical act. Molecular scissors and glue, cutting out a tiny piece of foreign DNA and stitching it into a specific location in the CRISPR array. Then, when the same phage returns, the bacterium uses that stored snippet to recognize it. Recognition is the first step of any immune system. Whether you are a microbe or a mammal, your immune system is loud. Swelling, fever, inflammation, a lot of expensive drama. The bacterial version is quiet, a matching sequence, a guided cut, a virus disassembled before it can speak. But we are not there yet in the timeline. At this point in the human story, the idea is still forming. People are still doing what people do best, arguing politely in journals, checking one another's work, trying to rule out coincidence. Because if you claim bacteria have an immune system based on genetic memory, you need more than vibes. You need experiments. You need to show that spacers change with infection. You need to show that adding a spacer changes resistance. You need to show that removing the system makes bacteria vulnerable. This takes time, not because scientists are slow, because molecules do not care about your grant deadlines. One of the key observations comes from bacteria used in food, like Streptococcus thermophilus. These are bacteria humans keep on purpose. They help make yogurt and cheese. They are domesticated microbes in a way, and like any domesticated creature, they still have predators. Phages that can ruin a fermentation batch. Industrial microbiology has a very practical interest in not losing entire tanks of product to viral attacks. So people watch these bacteria closely. They track phage resistance. They notice that resistant strains often have different CRISPR spacers. And when bacteria are exposed to new phages, sometimes new spacers appear, added into the array like a fresh clipping. And those new spacers match the phage that just tried to kill them. This is the kind of evidence that feels almost unfairly neat, like nature, for once, filled out the form correctly. Bacteria challenged with a phage acquire new spacers, become resistant, and the spacer matches the attacker. It is not magic. It is a system, a set of steps, and now the repeated sequences start to look less like nonsense and more like scaffolding. They are the uniform dividers in the scrapbook. They allow the cell to process the whole region into smaller pieces later. They provide structure, like perforations on a sheet of stamps. The spacers are the important parts, the remembered faces. But the repeats make it possible to handle the memory in a controlled way. If you are the bacterium, this is not nostalgia. This is inventory management. If you are the scientist, this is the moment your tired eyes widen just a little. Not because you are thrilled, because you are trying to stay skeptical. Because your job is to be the person in the room who says, prove it, even when you want it to be true. So you test it, you knock out CRISPR genes, and resistance collapses. You add spacers, and resistance appears. You observe that the system targets matching sequences in the invader, and you begin to see the outline of the mechanism. Not all the details yet, but enough to stop calling it weird repeats. Enough to start calling it immunity. A microbial immune system that uses genetic sequence recognition rather than antibodies. Now, if you let your mind drift into the drop of water again, you can picture what is happening in real time. A phage drifts close, like a small machine with one purpose. It docks. It injects. Inside the bacterium, the CRISPR region is not just sitting there. It can be transcribed into RNA, like copying a page of a record book. Then those transcripts are processed into smaller guide pieces. Each one contains a spacer sequence, a remembered snippet. These guide RNAs can pair with matching viral sequences, like two sides of a zipper. When they match, Cas proteins are recruited. Cas proteins are enzymes that cut nucleic acids, scissors in the simplest sense, and when they cut the viral genome, the infection fails. The virus cannot complete its replication. The bacterial cell continues its small routines, metabolizing, dividing, ignoring your need for meaning. It is almost comforting, the way it reduces survival to a matching problem. If the invader matches the memory, it gets cut. If it does not match, the bacterium may die. No outrage, no bargaining, just chemistry. Of course, viruses do not like being cut up. They mutate. They change the target sequence so the zipper does not close cleanly. They produce proteins that interfere with CRISPR systems. Yes, even viruses have countermeasures. Even here, in the dark water, there is an arms race. Not heroic, not cinematic, just endless. A quiet, efficient war for replication. And that war leaves fossils in genomes. Those spacer arrays are like little battlefield souvenirs, a record of who has come through, a record of what the cells survived long enough to remember. Now, you might be tempted to think the discovery was an instant revelation, a dramatic paper, a room full of applause. But it was mostly incremental. It was scattered across labs and years. Different groups naming pieces, different experiments confirming parts, a slow convergence of evidence. Science often looks like that, not a spotlight, more like dawn. And in that dawn, CRISPR shifts from curiosity to concept, a system with phases, adaptation, where new spacers are acquired, expression, where CRISPR RNA is made from the array, interference, where matching invader DNA is cut, and you, unlucky enough to be alive inside the model organism of the moment, get to feel what those phases mean in your cytoplasm. You do not feel them as words. Of course, you feel them as timing, as a sudden change in what your proteins are willing to do, as the difference between a normal night of metabolism and a night when foreign code arrives. You think of virus attacks like a predator with teeth. Wrong. It attacks like paperwork. A thin strand of nucleic acid slides in, and your enzymes start obeying it. Your ribosomes, those tireless bead-like machines, begin reading a script that is not yours. The phage does not need to shout. It only needs to be legible. Your cell wall is not a castle. It is a membrane under tension, a wet boundary, pretending to be a border. When the phage genome enters, it feels less like invasion and more like being edited. And you have, tucked away in your chromosome, a small library of edits you already hated once. The first step, the one humans call adaptation, is the least romantic. It is not revenge. It is not courage. It is filing, somewhere in your cell, proteins that humans eventually label Cas1 and Cas2 handle broken fragments of foreign DNA. They sample the intruder, not the whole thing, just a small piece. They choose a fragment like you might tear off the corner of a threatening letter, just enough to recognize it later. Then, with the calm confidence of chemistry, they insert that fragment into your CRISPR array, always near the same end, like adding a new entry at the top of a logbook. The repeats act like the ruled lines on the page. They make sure the new spacer lands in the correct frame. Congratulations! Your genome just took notes during a mugging. This is not guaranteed to happen every infection. Sometimes you die too fast to remember. Sometimes the phage is too efficient and your cell becomes a spray of nutrients in the water. But when you do survive, when the timing is right, you keep a piece. You make the attack part of your own genetic furniture. Humans had to work to prove this, because bacteria do not give interviews. They did it by watching spacer arrays change over generations. They did it by exposing bacteria to known phages, then sequencing the survivors. They saw new spacers appear like fresh scars aligned with the phage genome. They saw that if you give bacteria a phage, you sometimes get a new genetic receipt. And they saw that those receipts mattered. Now comes expression, the part that makes the library usable. Your CRISPR array is DNA, long-term storage. But when danger arrives, you need working copies, So you transcribe the array into a long RNA molecule, a single ribbon containing repeats and spacers in sequence. It is like photocopying the entire scrapbook in one long strip. And then you cut it up, not randomly, but at the repeats, because the repeats are designed to be recognizable. The repeats can fold into little hairpin shapes in RNA, small loops that act like handles. Those shapes help enzymes process the long transcript into individual guide pieces. Each piece, each small CRISPR RNA, carries one spacer, one memory, one address. In many systems, another RNA is involved too, a helper that pairs with the repeat region. Humans later call it transactivating CRISPR RNA, which is a long way to say the assistant that helps the guide get dressed. In your cell, none of this is fancy. It is just molecules bumping into the right partners often enough to become a system. Then comes interference, the part that decides whether you get to keep existing. A guide RNA floats through your cytoplasm, held in a Cas protein like a blade with a handle. It is not looking for beauty, it is looking for a match. When the phage genome is present, it has sequences that can pair with the guide letter for letter. If pairing happens, the Cas protein changes shape. That shape change is a decision in the only sense molecules can decide. And the decision is to cut. Different bacteria use different Cas proteins. Some target DNA, some target RNA. Some work as teams, building larger complexes that patrol like silent machines. But the core feeling is similar. Match, then damage. The phage genome is chopped, nicked, shredded, or stalled. Replication fails. Assembly fails. The factory shuts down before the first product leaves the line. From the outside, if you are a human watching a culture plate, it looks boring. Some bacteria live. Some do not. If you are the bacterium, it is still boring, but in a way you appreciate. Because the alternative is lysis. The alternative is becoming a brief cloud of carbon and phosphorus for someone else. Now, the part humans love to forget, because at CRISPR is not perfect. If the guide matches your own genome, you can cut yourself. Autoimmunity, but microbial, and with less poetry. So CRISPR systems often need safeguards. They look for small signals near the target, little motifs that help distinguish self from non-self. In some systems, the Cas proteins require a short neighboring sequence in the invader DNA, a protospacer adjacent motif, a tiny tag that usually is not present next to the stored spacer in your own array. It is a simple trick, not foolproof, but good enough for a bacterium that divides before sunrise. And of course, the phage has its own opinions. A phage can mutate the target sequence, changing a letter so the guide binds poorly. A phage can delete the region entirely, if it can afford it. A phage can even bring tools to sabotage your defenses. There are phage proteins that inhibit CRISPR, anti-CRISPR proteins, small molecular wrenches thrown into your cutting machinery. You build an immune system, and the universe immediately invents a counterexample. That is how evolution keeps itself entertained. In the lab, humans watched this arms race like a slow chess game played with invisible pieces. They saw that when CRISPR works, phages are pressured to change. They saw that when phages change, bacteria acquire new spacers they saw arrays expand like a timeline of conflict. Older spacers at one end, newer spacers at the other. A genetic diary written in alternating lines of repetition and panic. There is something grimly comforting about how literal it all is. A memory is a sequence. Recognition is base pairing. Defense is cleavage. If you are used to thinking of immunity as antibodies and fever, this feels like a different genre. It is immunity as text comparison, immunity as find and replace. Now, if you drift back into the human story, you can picture the moment the system stops being merely interesting. Because humans have a particular weakness. When they see a tool in nature, they immediately picture themselves holding it, not carefully at first, just imagining. The first wave was naming and sorting. Scientists cataloged different CRISPR systems because bacteria are incapable of standardization. They found different classes and types with different Cas proteins and architectures. Some systems use large complexes, many subunits assembled into a surveillance machine. Others use a single larger protein that can do the job alone. The single protein version is the kind humans love because it is simpler to repurpose. And this is where certain names begin to hover in the background, like quiet characters waiting for their entrance. Cas9, Cas12, Cas13. Different tools for different jobs, all born from bacterial desperation. But before any of that becomes a technology headline, it begins as a question asked in a calm room. Can you program it? Can you change the guide and make it cut somewhere else? If the guide is just sequence, then in principle, you can write your own memory. Not by surviving an infection, but by designing an RNA that points to any target you choose. In other words, can you turn bacterial immunity into a general-purpose editing system? The early demonstrations were not flashy. They were the kind of experiments that look like someone verifying a lock works by locking and unlocking it. Researchers showed that a Cas protein could be guided to a target sequence in a test tube. They showed that changing the guide changed the target. They showed, slowly, carefully, that this was not a coincidence. The system was not mystical. It was programmable. And once something is programmable, humans begin to treat it like software, with all the confidence and all the consequences that implies. There is an important detail here, and it is easy to miss because it sounds too simple. The guide RNA is not the scissors, it is the address label. The Cas protein is not the address, it is the blade. And when you bring them together, you get a package that delivers a cut to a chosen location. If you are editing text, a cut is just a deletion. In a cell, a cut is an emergency. DNA does not like being broken. When a double-strand break occurs, the cell scrambles to repair it. It uses pathways with names that sound polite but behave like hurried repair crews. Non-homologous end joining is one. It often slaps the broken ends together quickly, sometimes losing or adding a few bases in the process. This is useful if your goal is to disrupt a gene, to create a small error that breaks the meaning of the sequence. Another pathway is homology-directed repair. This one can use a template, a matching sequence, to repair the break more accurately. If you provide a template with a desired change, the cell can copy it into the genome. Can is doing a lot of work in that sentence because cells are not vending machines. They are busy, moody chemical cities, and repair does not always go the way you want, but the concept is clear enough to be dangerous. If you can cut a gene, you can knock it out. If you can cut and provide a template, you can change it. You can fix a typo, or introduce one, or rewrite a paragraph. And once humans realized this, the story accelerated not in tone, but in consequence. You can picture the lab benches where this happened. Not grand cathedrals of knowledge, just cluttered surfaces with pipette-tip boxes and coffee stains. Graduate students moving quietly because it is late and the incubators do not care. They build plasmids, circular DNA tools that can carry the Cas gene and the guide RNA into cells. They transfect cells in dishes, mammalian cells that look like pale islands. Under a microscope, they wait. They extract DNA. They amplify the target region with polymerase chain reaction, that repetitive copying trick biology already uses. Then they sequence, and they see the edit, a small change, like a comma moved in a sentence. That is the moment where bacterial survival machinery becomes human technique. Not because it wanted to help, because it did not get a choice. There is a temptation to talk about this as if humans invented something. They did not. They discovered a knife already in nature's drawer, and they learned how to hold it by the handle. And then, because humans are humans, they immediately started imagining what else could be sliced. In agriculture, they picture crops with resistance to disease. In medicine, they picture therapies that correct mutations. In basic research, they picture turning genes off and on, like lights in a building. And in quieter corners of the mind, they picture edits that are not just therapeutic, edits that alter inheritance, edits that change what future humans are allowed to be. But those thoughts are for later. For now, stay in the lab, where the work is still mostly intentionally boring. Because the early uses of CRISPR in human hands are not about redesigning life. They are about understanding it. If you want to know what a gene does, one of the cleanest ways is to break it. Not philosophically, literally. You remove its function and you watch what fails. Cells stop dividing. A protein disappears. A pathway stalls. A mouse develops differently. It is crude in a way, like pulling fuses to map an electrical system. But it is effective. Before CRISPR, gene editing was possible, but it was slower and more expensive. Tools like zinc finger nucleases and transcription activator like effector nucleases existed, and they were impressive. They were also finicky, like custom-made keys that took too long to file. CRISPR made targeting easier because changing the guide RNA is simpler than re-engineering a whole protein. You write a new address label, and the same scissors can be reused. This is why the technique spread so quickly. Not because it was trendy, but because it was practical. Science, at its core, is a search for methods that reduce suffering, including the suffering of the person doing the experiment. Now, if you are the viewer in bed, you might be thinking of gene editing as a clean, precise thing. A surgeon's scalpel, a single cut, a single fix. But the cellular reality is messier. Even with a well-designed guide, the Cas protein can sometimes cut similar sequences elsewhere. Off-target effects, they call it, which is a gentle phrase for unintended damage. It is like telling someone to remove one typo from a document, and they also delete a few random letters in the margins. Sometimes, those changes do nothing noticeable. Sometimes, they matter. So researchers learn to measure and minimize off-target edits. They engineered improved Cas proteins with higher fidelity. They designed guides more carefully. They used computational tools to predict risky matches, because when you are cutting genomes, you do not get to be casual. Cas proteins themselves have quirks. Some require that little motif near the target sequence. That constraint can be annoying, like a lock that only opens if the door has a specific sticker on it. But it also adds specificity, which can be useful. Different Cas enzymes recognize different motifs, which means some are better for certain targets than others, and some cut differently. Cas9 typically makes a blunt cut, both DNA strands near the same position. Cas12 often makes staggered cuts, leaving overhangs. These details matter when you are trying to guide repair. In the quiet hours, researchers compare results, like bakers comparing ovens. Same recipe, different heat, different texture. And they keep iterating, because the goal is not just to cut. The goal is to control what happens after the cut. That is always the hard part. Cells repair DNA in ways optimized for survival, not for your experiment. They will choose the fastest repair path available. They will introduce small insertions or deletions. They will surprise you, because biology is a machine built from compromises. This is why newer approaches emerged, still under the umbrella of CRISPR, but less dependent on violent breaks. Base editing is one of them. Instead of cutting both strands, you bring a modified enzyme that can change one base to another, like converting a letter without ripping the page. A cytosine can become a thymine. An adenine can become a guanine. It is still chemistry, still precise in concept, but gentler in execution. Then came prime editing, a more complex method that uses a Cas protein nickase and a reverse transcriptase. It can write new information into the genome with fewer double-strand breaks. It is like a molecular word processor, still imperfect, but capable of more nuanced edits. These tools are often described as upgrades, but they are really adaptations. Humans trying to make the system behave less like bacterial warfare and more like delicate surgery. Because bacteria do not care if the repair is messy, as long as the invader dies. Humans, unfortunately, care. Now, zoom out slightly, not to the cosmos, but to the broader microbial world. CRISPR is not the only defense bacteria have. It is one of several strategies in a long history of microbial paranoia. Before CRISPR was understood, scientists already knew about restriction enzymes. Those are proteins that cut DNA at specific sequences, like molecular paper shredders. They are part of restriction modification systems, where the bacterium protects its own DNA by methylating it, tagging it, while foreign DNA lacks that tag and gets cut. This is an older kind of immunity, more general, less memory-based. If CRISPR is a scrapbook of specific enemies, restriction enzymes are a bouncer checking for the right wristband. And bacteria have more tricks. Some use abortive infection systems, a kind of cellular self-destruct. If infection is detected, the cell sacrifices itself to stop the phage from reproducing. A grim altruism, if you want to anthropomorphize. Or just a genetic strategy that benefits the colony, because bacteria often live in communities where kin share genes. Some bacteria alter their surface receptors, making it harder for phages to attach, like changing the locks every night. Others produce extracellular matrices slimy biofilms that act as physical barriers. The microbial world is not a peaceful soup. It is a landscape of fortifications. And CRISPR is one of the more elegant ones because it learns, it records, it adapts. That learning is not conscious, but it is still information stored through time. And humans, seeing that, could not help but recognize a theme they love. The theme of memory. The theme of past harm shaping future response. The theme that even simple life, made of membranes and enzymes, carries history. In the human timeline, key papers accumulated like stones in a riverbed. By the early 2000s, CRISPR was named. By the mid-2000s, spacer matches to phages were noticed. Around the late 2000s, experiments showed new spacer acquisition after phage challenge and the link to resistance. Then, in the early 2010s, the programmable cutting potential crystallized. A few groups demonstrated that a Cas protein guided by RNA could cut chosen DNA sequences. And soon after, it was shown in eukaryotic cells, the kind with nuclei, the kind humans are made of. At that point, the system was no longer only a microbial story. It became a human story too, with all the noise that brings. But keep your attention on the quiet parts, because the quiet parts are where the science lives. The careful cloning steps, the controls, the repeated measurements, the nights where nothing works because the guide was poorly designed, or the cells were unhappy, or the transfection reagent degraded. The reality of CRISPR in a lab is not a cinematic montage. It is a person labeling tubes with a marker that is running out of ink. It is a gel that shows smears instead of bands. It is a sequence file that looks like nonsense until you align it correctly. And then, slowly, it becomes clear. Clear, usually, means you have finally asked the right question and the molecules have stopped punishing you for it. Now the next question arrives, soft and heavy, like a lab door closing without a click. Once you can aim the scissors, what do you actually do with them? You might think the first answer is dramatic, like curing everything in a week. Wrong. The first answer is that you cut things on purpose just to see what breaks, because biology is not a neatly labeled machine. It is a heap of parts that mostly works, and nobody included the manual. So you start with the most boring kind of bravery. You take a gene that you suspect matters. You design a guide RNA, a small strip of letters that points like a finger. You put Cas and the guide into cells. Then you wait, and you check, and you count, and you discover the same truth repeatedly. Cells do not care what you suspect. They care what happens. In a dish, a cell line looks calm. A thin sheet of living islands, pale and slightly annoyed. But inside, it is constant motion. DNA is packed and unpacked. RNA is made and destroyed. Proteins fold, misfold, and get hauled away like trash. So, when you introduce CRISPR, you are not giving the cell a gentle suggestion. You are introducing a controlled emergency, a targeted break, and then the cell does what it always does. It repairs quickly, imperfectly, and for its own reasons. If you are trying to knock out a gene, you are hoping for a repair that makes a mess. A small insertion or deletion, a few letters lost or gained, just enough to shift the reading frame, like misaligning the subtitles. Congratulations, your gene is now speaking nonsense. That is often the goal, and when the gene stops working, you watch the consequences. Does the cell stop dividing? Does it change shape? Does it fail to respond to a signal? Does it die quietly or keep going like nothing happened? This is how you map function, not by admiring the genome, but by vandalizing it with consent. But humans are never satisfied with only breaking things. They also want to fix. And fixing requires a more delicate bargain with the cell. If you cut DNA and provide a template, the cell may copy from that template. It may, on a good day, incorporate the change you want. On a bad day, it ignores your template and does whatever is fastest. Because in the hierarchy of needs, accuracy is a luxury. Survival is the priority. So researchers learned to work with timing. They learned which phases of the cell cycle favor careful repair. They learned to deliver templates in different forms: single-stranded DNA, double-stranded DNA, longer cassettes. They learned that the size of the edit matters, like trying to patch a tiny tear versus replacing a whole sleeve. They learned to accept partial success, because biology does not do perfection. It does distribution curves. If you edit 1,000 cells, you might get a few with the exact change you wanted, a few with messy repairs, a few untouched, and you have to separate them, isolate them, confirm them. This is why gene editing is both powerful and slow. The cut is quick. The certainty is expensive. Now drift to another bench in the same dim lab, where the work feels almost like accounting. You are not editing one gene in one dish. You are editing many genes in many wells and keeping track. This is where CRISPR becomes less like a scalpel and more like a printing press. You can build libraries of guide RNAs, thousands of different guides, each targeting a different gene. You deliver them into a population of cells, so each cell gets one guide, like a random assignment. Then you apply pressure, a drug, a toxin, a virus, a low-nutrient condition, and you ask a quiet, powerful question. Which edits let cells survive? Which edits make them disappear? You read out the answer by sequencing the guides remaining in the population. The guides are barcodes. The survivors carry the evidence. This is called a CRISPR screen, and it is one of the most practical uses of the system. Not because it is glamorous, because it lets you find genes involved in a process without guessing. If you want to know what helps a cancer cell resist a drug, you can screen. If you want to know what genes a virus depends on to enter a cell, you can screen. If you want to know what makes a neuron fragile or a stem cell stable, you can screen. The results are not always clean. Some genes matter only in certain contexts. Some edits have subtle effects. Some cells cheat, and some guides work better than others. But compared to older methods, this feels like switching from candlelight to a desk lamp. Same room, less suffering. And while you are doing all this, in the background, you begin to notice a second, quieter twist. You do not actually need to cut DNA to make CRISPR useful, because Cas proteins can be modified. If you change key amino acids, you can make a Cas that still binds DNA, but does not cut it. A dead Cas, they call it, with the kind of casual cruelty scientists reserve for proteins. Dead Cas still listens to the guide RNA. It still goes to the target sequence. It simply sits there, occupying space, blocking transcription like a parked truck. This becomes CRISPR interference. a way to turn genes down without breaking them. And if you fuse that dead Cas to an activator, you can turn genes up. Now you are not editing the DNA sequence, you are editing gene expression, the volume knob. You can suppress, you can enhance, you can perturb networks gently without ripping the page. This is why the system spread through biology so quickly. It is not one tool. It is a platform, a targeting mechanism that can carry different payloads. Scissors, yes, but also tape, also highlighters, also little flags you can stick onto the genome to recruit other proteins. And you keep discovering new variations because bacteria have been inventing them for billions of years. Humans are just taking notes. Now, speaking of notes, there is a detail in the original bacterial system that is easy to forget in the human version. Bacteria are not editing themselves for beauty. They are fighting. CRISPR in nature is not a clean laboratory trick. It is an immune strategy, and it comes with collateral damage. If you are a bacterium, acquiring new spacers is risky. You might accidentally capture DNA from something that is not a phage. A plasmid? Maybe? A small DNA circle? That could be helpful. Plasmids often carry useful genes, like antibiotic resistance. So CRISPR can protect you from viruses and also prevent you from acquiring helpful traits. There is a trade-off, quiet and constant, security versus opportunity. And that trade-off shows up in genomes. Some bacteria have CRISPR systems that are active and large. Some have remnants, degraded repeats, broken Cas genes. Some seem to have lost CRISPR entirely, perhaps because in their niche it was more trouble than it was worth. Because immunity is not free. It costs energy. It costs complexity. It costs mistakes. Humans have the same problem, just with different paperwork. So when you see a bacterium without CRISPR, do not assume it is defenseless. It may have other systems, older ones, stranger ones, systems that look like sabotage. Restriction enzymes? Yes, those blunt bouncers, but also toxin-antitoxin systems, little paired genes where one makes poison and the other makes the antidote. If a phage disrupts the balance, the poison wins and the cell self-destructs. It is a dead man switch built out of protein. There are also systems with names that sound like someone got tired halfway through writing them. Defense Islands, Brex Disarm, and other acronyms that hide real complexity. The microbial world is not a single clever trick. It is a whole arsenal of grim options. And phages respond with their own arsenal. Anti-CRISPR proteins are just one example. Some phages modify their DNA chemically, so it is harder to recognize. Some hide inside bacterial genomes as prophages, lying dormant, becoming part of the host until conditions change. Some carry genes that help bacteria compete, bribing their host into keeping them around. This is what makes the idea of microbial life so quietly unsettling. Even at this scale, nothing is purely good or purely bad. Everything is conditional. Everything is negotiated with survival. Now, come back to the human use, because this is where your species gets restless. Once you can edit genes in cells, you ask the next question. Can you do it in living organisms? In plants? In animals? This is where the story stops being a neat lab technique and becomes logistics, because editing in a dish is one thing. Delivering the machinery into the right cells in a body is another. You need to get Cas and guide into tissues without provoking chaos. You need to avoid immune reactions. You need to control where the edit happens. You need to control how many cells are edited. You need to make sure the edit is the one you intended, and the body is not a gentle environment for foreign packages. It is full of enzymes that chew up RNA. It is full of immune cells that treat unfamiliar proteins like a fire alarm. So delivery becomes the central problem, not the cutting. Getting there. Some approaches use viruses, ironically, because viruses are good at entering cells. That is their entire personality. So researchers use viral vectors like adeno-associated virus, engineered to be less dangerous. They pack the gene for Cas and the guide into the vector. They inject it, they hope it reaches the right tissue. But viruses have cargo limits. Some Cas proteins are too large to fit easily. So people choose smaller enzymes, or split the cargo, or use different delivery systems. Other approaches use lipid nanoparticles, little fat bubbles that can carry RNA. They merge with cell membranes, delivering instructions like a quiet envelope. This is similar in spirit to some modern vaccines, which taught the public, briefly, that RNA can be medicine, a fleeting moment of collective learning before everyone returned to their usual hobbies. With lipid delivery, you can deliver messenger RNA for Cas and the guide RNA. The cell makes the Cas protein temporarily, does the edit, and then the instructions decay. Transient exposure can reduce long-term risk, but deliver is still uneven. Some tissues are easy to reach, like the liver. Some are harder, like the brain. And some are easy to reach but ethically complex, like reproductive cells. You see how the problems pile up, not like drama, but like forms that need signing. Now, because this is bedtime science, we stay with the softer practical uses first. CRISPR in medicine is already being used, carefully, in specific contexts. One early path was editing immune cells outside the body, ex vivo, meaning you take cells out, edit them, and put them back. This gives you control. You can check the cells before reinfusion. You can measure edits. You can expand the cells that look right. It is still complex, but it is less like throwing scissors into a crowd. A notable example involves sickle cell disease, a single letter change in a gene for hemoglobin, leading red blood cells to deform. Those cells become stiff and sickle-shaped, clogging vessels, causing pain and damage. The disease is simple in its genetic cause and brutal in its biological effect. CRISPR-based therapies have targeted this in clever ways, not always by fixing the original mutation directly, but by changing regulation. For instance, by editing a regulatory region to reactivate fetal hemoglobin, a form used before birth. Fetal hemoglobin can compensate for the faulty adult version, so you do not correct the typo. You open an older chapter that the body usually closes. It is a workaround, which is what medicine often is. Because the body is not a code base you wrote. It is inherited legacy software with no comments. There are also efforts in eye diseases, where local delivery is possible. If you inject editing machinery into the retina, You can target cells directly. Again, this is not casual, but it is feasible in a way that whole body editing is not. Cancer research uses CRISPR in another way, by editing T cells, immune cells that can be trained to attack tumors. You can remove inhibitory receptors. You can add synthetic receptors. You can change how the cell recognizes targets. Some of this uses CRISPR. Some uses other genetic methods. But the idea is similar. You are rewiring the immune system, which is already a weapon, to point at a different threat. Congratulations. You are now customizing biology like it is a device. And, like all devices, it can malfunction. So, the work proceeds with caution, and with monitoring, and with the awareness that bodies are not test tubes. Now, shift from medicine to agriculture, where the editing feels less intimate but affects more mouths. Plants are edited with CRISPR to alter traits, disease resistance, drought tolerance, shelf life, nutritional profiles. Sometimes you knock out a gene that makes a plant susceptible to a pathogen. Sometimes you tweak regulation to change when fruit ripens. Sometimes you remove a gene that produces a bitter compound. It can be subtle, like turning down a flavor note. And because CRISPR can make changes without necessarily introducing foreign DNA into the final plant, it complicates regulatory categories. Traditional genetic modification often involved inserting genes from other organisms. CRISPR can make edits that could, in principle, arise naturally. A deletion, a point mutation, a small change in expression. This has led to debates about how to regulate edited crops, not because the molecules care, because humans do. Humans care about labels and trade and trust is slower to engineer than DNA. In animal research, CRISPR made it easier to create model organisms. Mice with specific mutations, zebrafish with fluorescent markers, cells engineered to report when a pathway turns on. This accelerates basic biology, which is not a headline, but it is the foundation under the headlines. When you hear that a disease gene was identified, there is often a trail behind it. Screens, knockouts, careful replication. CRISPR made that trail shorter, not effortless, shorter. Now, the soft thought that follows naturally is also the one people whisper about late at night. If you can edit somatic cells, the cells of an individual, that affects only that person. If you edit germline cells, sperm, eggs, embryos, then the edit can be inherited. It passes forward, quietly, into people who did not consent. This distinction matters. Somatic editing is like repairing one house. Germline editing is like changing the blueprint for every future house in the neighborhood. Sometimes that sounds appealing if you are thinking about eliminating genetic diseases. Sometimes it sounds terrifying if you are thinking about misuse, inequality, and irreversible mistakes. And because humans can turn any tool into a status symbol, the concern is not theoretical. The world already has enough systems for making some lives easier than others. Gene editing could become another lever. So ethics enters the story, not as a dramatic villain, but as guardrails, rules written in committees, debated in conferences, revised after mistakes. Ethics is not there to stop science. It is there to slow it down to a speed the consequences can keep up with. If you have ever regretted a message you sent too fast, you understand the principle. Now, there was a moment in the real world that made these questions less abstract. A researcher announced the birth of edited babies, claiming to have altered a gene related to HIV susceptibility. The details were widely criticized. The consent process, the necessity, the safety, the oversight. It was a case study in what happens when someone treats a powerful method like a personal project. The scientific community responded with condemnation and with renewed calls for regulation, because even if the edit had worked exactly as intended, which is not clear, the act crossed lines. Not just technical lines, social ones, moral ones. And it reminded everyone that the limiting factor is not always the technology. Sometimes the limiting factor is human judgment, which is famously unreliable at 2 a.m. But the ethical conversation is not only about extreme cases, it is also about quieter, ongoing decisions. What diseases justify risk? What counts as therapy versus enhancement? Who gets access? Who bears the risk? How do you run clinical trials responsibly? How do you monitor long-term effects? How do you account for genetic diversity across populations so benefits are not limited to those already studied? These questions are not answered once. They are revisited, like checking a lock before bed. Because the environment changes, the tools improve, the incentives shift, and the only stable truth is that people are capable of both care and recklessness. Now, if you pull back a little, you can see why CRISPR provoked such attention. It made gene editing more accessible. It lowered barriers. It moved capability from a few specialized labs to many. That is good for discovery. It is also a reason to take safety seriously, not because everyone is malicious, because accidents scale with participation. Most harm in science is not sabotage. It is error, misunderstanding, haste. So laboratories adopt best practices. They validate guides, they sequence off-target sites, they use controls, they share protocols. They publish limitations alongside successes, boring habits, the kind that do not trend, the kind that keep the floor from collapsing. Now, speaking of habits, you should remember that CRISPR did not appear in human hands as a single clean story. There were disputes, patents, credit assignments, because whenever something becomes valuable, humans begin to argue about who saw it first. This is not unique to CRISPR. It is a recurring feature of your species. A tool can be born from bacterial survival and still end up in courtrooms. The science continues anyway. The bacteria do not care who gets the prize. They keep fighting phages in the dark, storing spacers like little receipts of violence. And in that microbial world, there are still details humans are learning. For example, spacer acquisition is not random. Some systems prefer certain protospacer adjacent motifs. Some have mechanisms called priming, where an imperfect match can trigger acquisition of new spacers from the same invader. Priming is the bacterium noticing a burglar with a slightly different hat and deciding to update the security camera anyway. It is not perfect recognition. It is recognition with suspicion. The guide RNA pairs badly, a few letters off, like a key that almost fits. But that near miss can still wake up the system. Instead of cutting cleanly and ending the story, the cell starts sampling more DNA from the same invader. More clippings for the scrapbook, more spacers added near the front of the array, like fresh entries in a logbook. In human terms, it is the way a bad experience makes you over-prepare next time. In bacterial terms, it is chemistry learning to be paranoid. And paranoia, in the microbial world, is often just another word for longevity. Now picture how cramped your life is. If you are the bacterium, you are a membrane stretched tight around salty water. You are enzymes bumping into each other in the dark. You are a single chromosome folded like a sleeping animal, and yet you are expected to store a history of enemies. You cannot store everything. Your genome is not a museum. So CRISPR arrays grow, and they also shrink. Spacers can be lost. Arrays can be rearranged. Older memories can be deleted, not out of forgiveness, but out of lack of space. The cell keeps what is recent, what is useful, what has been tested. Everything else becomes genetic compost. This is one reason the arrays can be used like timelines. If you sequence the CRISPR region in bacteria from the same place, you can sometimes infer who encountered what and when. It is not a diary with dates. It is a stack of receipts in the order they were crumpled into a pocket. Still, it tells you something. It tells you the water was dangerous. It tells you which phages were common enough to matter. It tells you survival has a signature. This is the part where you might expect bacteria to be neat about it. You might imagine a tidy immune archive, carefully curated. Wrong. The bacterial world is full of messy compromises. Some CRISPR systems target DNA. Some target RNA. Some rely on large protein complexes that patrol slowly. Some use a single enzyme that does most of the work alone. Even the repeats differ because evolution is allergic to standardization. So when humans talk about CRISPR as if it is one thing, they are simplifying, which is fine. You are trying to fall asleep, not defend a thesis committee. But the underlying truth is that CRISPR is a family of systems, all built around the same idea. Store a snippet, make an RNA copy, use it to find a match, then do something unpleasant to the match. And even that shared idea hides more detail, because the cell has to avoid targeting itself, which is an easy mistake to make. A spacer is taken from an invader, but the spacer is stored in the genome. So later, when guide RNAs are made, they contain sequences that exist inside the cell. If Cas proteins attacked anything that matched the guide, the cell would commit molecular self-harm constantly. So, the system needs a way to tell stored memory from active invader. That is where the little neighboring tag matters so much. That short motif, the protospacer adjacent motif, is like a stamp that says, this sequence came from somewhere else. The bacterial genome has the spacer sequence, yes, but it lacks the stamp next to it in the CRISPR array. So the Cas protein looks for both things, a matching sequence and the correct nearby stamp. Memory plus context. That is how you avoid cutting your own book while searching for a quote. And of course, phages learn this too. They evolve away from recognizable stamps. They shift their sequence so the motif is less common. Or they mutate the target region so the guide binds weakly. The match becomes fuzzy, like a face you almost recognize in bad light. And you, the bacterium, respond by capturing new spacers that target new regions. The arms race continues in small, quiet steps, with no winners that last. If you have ever wondered why microbes evolve so quickly, this is part of it. A phage can replicate in minutes. A bacterium can divide in minutes. Selection is not a slow philosophical pressure here. It is immediate. The water is full of killers, and the losers dissolve into nutrients. Now, let your imagination drift down into that drop of seawater again. You are not a scientist now. You are a single bacterium in a crowded darkness. Around you, there is salt and organic debris and other cells exhaling waste into the shared environment. There is no up or down, only currents. And above all, there is contact. Tiny collisions, constant, unavoidable. A phage bumps you like a drifting seed pod. It sticks, it injects. Foreign DNA enters your cytoplasm, and the first sensation is not pain, because you do not do pain. The first sensation is misallocation. Your ribosomes begin to translate instructions that do not benefit you. Your nucleotides begin to disappear into copies of something else. Your energy is diverted. It is like waking up and discovering your entire day has been scheduled by someone you hate. If your CRISPR system has a matching spacer, the response is quick. Guide RNAs are already present or are made rapidly, depending on the system. Cas proteins bind the guides and begin their patrol. Patrol is a metaphor, but it fits the feeling. These complexes drift through the cytoplasm, bumping into nucleic acids, testing matches the way a tongue tests a sore tooth. When they find the invader sequence with the correct stamp, binding becomes stable. The Cas protein changes shape, like a tool unfolding, and then the cut happens. A precise break in the phage genome, followed by other enzymes chewing up the fragments. The infection collapses. The phage has instructions, but no intact script. It cannot complete the takeover. Inside you, metabolism rebalances. The panic ends without a sound. From the outside, no one would notice anything happened. That is the comfort and the horror of microbial life. Catastrophe resolved in silence. But sometimes the match is imperfect. The phage is similar to one you have seen before, but not identical. The guide binds loosely, and the cut is delayed or incomplete. This is where priming can matter. The system is alerted, not confident. It begins grabbing new fragments from the invader DNA, adding them as new spacers. You are updating your defenses mid-attack. It is like writing down a license plate number while the car is still speeding away. Sometimes you survive long enough to finish the note. Sometimes you do not. If you do survive, the descendants of your cell will carry that new spacer, a molecular story told forward. Now drift back up, out of the water, into the human world again. Because humans did not just discover that CRISPR exists. They discovered that it is diverse, and that diversity is useful. Different Cas enzymes became different tools. Cas9 became famous because it could be guided by a relatively simple RNA, and it made a clean double-strand break in DNA. Cas12 became interesting because it cuts differently and recognizes different target stamps. Cas13 became a different kind of favorite because it targets RNA, not DNA. That matters because RNA is temporary. If you cut RNA, you change gene expression without changing inheritance. It is editing that fades like footprints in sand. And along the way, researchers noticed something else. Some Cas proteins do not just cut their target. Once activated, they begin cutting other nearby nucleic acids as well. Collateral cutting, a kind of molecular frenzy. In bacteria, this can be part of defense, turning infection into a broader shutdown. In human hands, this quirk became a feature. It became the basis for diagnostic tests. If a Cas enzyme cuts a target sequence, it activates, and then it cuts a reporter molecule that produces a signal. So you can detect a virus not by culturing it, but by sensing its genetic material. A quiet click of recognition turned into a readout you can see. This is how a microbial war trick becomes a medical test on a countertop. Life is full of these uncomfortable reuses. Now, you might think that once humans learned to use CRISPR, the story would be mostly engineering. And it is, partly, but it is also still discovery. Because bacteria and phages are still teaching us new rules. For example, many bacteria carry CRISPR systems that are inactive or partially broken. Sometimes the Cas genes have mutations. Sometimes the repeats degrade. Sometimes the array exists without the machinery to use it. Why keep a dead system? Because genomes are messy, and removal is not always clean. Because a system might be dormant, not dead, activated only in certain conditions. Because pieces can be repurposed. Because evolution is a hoarder. And sometimes those dead-looking systems still matter in subtle ways. They can affect gene regulation. They can influence how bacteria interact with plasmids. They can shape microbial populations, even when they are not actively cutting phages. The more you look, the less simple the story becomes. That is the usual outcome of looking. Another detail is that bacteria do not live alone. They live in communities, in biofilms, in guts, in soil, in oceans, in places where DNA is shared. Horizontal gene transfer is common. Genes move sideways between organisms, not just down through offspring. Plasmids carry cargo like smugglers. Viruses move genes too, sometimes accidentally packaging bacterial DNA and delivering it to other bacteria. So, CRISPR is not only about defense, it is also about controlling what foreign DNA gets to enter your life. And that has social consequences. In microbial terms, if you block plasmids, you might lose access to useful traits like antibiotic resistance genes or metabolic tools. If you allow plasmids, you might gain flexibility, but also invite phage-like elements. So CRISPR can shape the evolution of bacterial communities by influencing gene flow. It is immune memory, yes. It is also border policy. And border policy always has unintended effects. Now, speaking of unintended effects, humans met their own version of that problem the moment CRISPR entered living bodies. Not just off-target cuts, but mosaicism. If you edit an embryo, not every cell may receive the edit at the same time, so the organism can develop as a patchwork. Some cells edited, some not. Some edited one way, some another way. A body becomes a mixed population, like a forest with different species of trees. In somatic therapy, mosaicism can be acceptable, depending on the disease. In germline editing, it becomes another layer of unpredictability, and unpredictability is the thing you never want to inherit. This is why the ethical guardrails matter so much. Not because people are fragile, because biology is complicated. Now let the tone stay soft, but the facts stay present. When humans consider gene editing, they divide it into categories. Somatic editing affects only the treated individual. Germline editing affects future generations. There is also another category that feels less dramatic, but matters just as much. Editing in the lab for research in cells that will never become a person. That is where most CRISPR work lives. In plates, in flasks, in carefully labeled tubes. The boring middle. And that boring middle is what builds the knowledge required for therapies to exist at all. You do not jump from discovering a bacterial immune system to safely editing human genes. You crawl. You crawl through control experiments, off-target mapping, delivery trials, toxicity studies. You crawl through the reality that cells in a dish are not cells in a body. You crawl through the immune system, which may recognize Cas proteins as foreign because they come from bacteria. Some humans have pre-existing immunity to certain Cas enzymes because their bodies have met the bacteria those enzymes come from. Your immune system remembers too, but with antibodies and T cells, not spacers. So, if you inject a CRISPR therapy, the body might treat the Cas protein like an invader. That can reduce effectiveness. It can also create risk. So researchers explore different enzymes from different microbes, hoping to find versions less likely to trigger immunity. Or they deliver the system in ways that minimize exposure. Or they edit cells outside the body and return them. All of this is slow work. Slow is good. Here. Now, if you have been listening long enough, you may have noticed a pattern. CRISPR begins as bacterial memory. Humans turn it into scissors. Then they try to turn scissors into a pen. They try to write, not just cut, base editors, prime editors, transcription control systems, diagnostics. Each one is a variation on the same theme, a programmable targeting mechanism that brings a molecular function to a chosen sequence. And that theme expands beyond DNA. You can target epigenetic marks, chemical tags that alter how genes are expressed without changing the sequence. You can recruit enzymes that methylate DNA or remove methylation. You can modify histones, the proteins DNA wraps around. You can change which parts of the genome are accessible and which are silent. This is not editing the letters. It is editing the way the book is shelved and read, and it matters because many diseases involve regulation, not just mutation. The sequence can be normal, but the expression is wrong. So CRISPR becomes a way to probe regulation, to test causality. If you silence and enhance a region and a gene turns down, you learn something. If you activate a promoter and a cell changes state, you learn something. This is the research use that rarely makes headlines, but it is the bulk of the real work. Now, speaking of silent regions, return for a moment to the bacterial genome. Because bacteria also have regulation, and CRISPR systems can interact with it. Some CRISPR arrays can target not phages, but the bacteria's own genes, shaping behavior. Some bacteria use CRISPR to control which mobile elements they keep. Some even use CRISPR like a weapon against other bacteria, targeting DNA delivered during competition. It is not common in all species, but it exists. The immune system becomes, in some cases, a tool for dominance. Congratulations, even microbes can be political. And microbes are very old politicians. Now let the story soften into something more grounded again. A lab at night. A researcher looking at sequencing results. Not for a human therapy, not for a headline, but for a simple pattern. A new spacer in a bacterial population. A shift in the CRISPR array that correlates with a phage outbreak in a fermentation tank, because one of the places CRISPR was understood early and clearly was in industrial microbiology, where microbes are money. In yogurt cultures and cheese starter strains, phage infection is not an abstract ecological event. It is lost product. So companies tracked phage resistance carefully. They selected strains with certain CRISPR profiles. They rotated cultures. They treated the microbial world like agriculture, with pests and defenses. And CRISPR became part of that practical toolkit long before the public learned the name. In those settings, CRISPR is not about editing a human embryo. It is about keeping milk from turning into expensive disappointment. That is the gritty truth of how science becomes useful. Often, the first use is boring and commercial. It pays for the rest. Now, if you want to understand the quiet ethics of CRISPR, it helps to understand the difference between what is possible and what is reliable. Possibility is cheap. Reliability is the thing you spend decades earning. You can make a cut in DNA fairly easily if you can deliver the machinery, but controlling the outcome of the repair is harder. And ensuring that no other cuts happen is harder. And ensuring that the edit does not create a new problem is harder still. Genes do not act alone. They are part of networks. So when you change one gene, you can ripple through pathways you did not intend to touch. Sometimes that is fine. Sometimes it is the point. Sometimes it is catastrophic, but only in a subtle way. Years later. This is why clinical trials take time, not because regulators enjoy paperwork, though some probably do. Because biology can hide consequences. You can edit a cell, and it can look healthy, and then behave differently after many divisions. You can create a rare off-target mutation that matters only in a specific context. You can alter the balance of growth and restraint in a cell, which is a polite way of describing cancer risk. So a therapy is not just an edit. It is an edit plus follow-up, plus monitoring, plus understanding. And understanding is usually the slow part. Now, ethics enters not as a single debate, but as layers of practice, institutional review boards, ethics committees, funding agency requirements, journal policies, international guidelines, not perfect shields, but guardrails. People discuss what counts as acceptable risk. They discuss informed consent, which is difficult when the science is complex. They discuss access, because a therapy that only wealthy people can afford becomes another inequality engine. They discuss cultural perspectives on disability, because not everyone agrees that every genetic condition should be eliminated. They discuss unintended consequences for ecosystems when editing is proposed in wild populations. And yes, they also discuss the fear that someone will try to create designer children. Fear can be melodramatic, but it can also be a signal. It tells you where consequences might be irreversible. Now, speaking of ecosystems, there is a particular CRISPR-related idea that sounds like science fiction and behaves like ecology. Gene drives. A gene drive is a genetic system designed to spread a particular trait through a population faster than normal inheritance would allow. Normally, a gene has about a 50% chance of being passed on from a parent. With a gene drive, that probability can be biased upward. The drive copies itself onto the other chromosome, turning heterozygotes into homozygotes. The result is that the trait spreads rapidly through a population. CRISPR makes building gene drives easier because it can provide the cutting and copying mechanism. People have proposed gene drives for controlling disease vectors, like mosquitoes that transmit malaria. The idea is to spread infertility genes, or genes that reduce the ability to carry parasites. It is a tempting idea. It is also terrifying in the quiet way, because once released, a gene drive can be hard to recall. Ecologies are interconnected, and removing a mosquito species or altering it can ripple outward. Predators, competitors, food webs. Also, borders do not stop mosquitoes. So a decision in one place can affect another place without asking. And that is why when people talk about gene drives, they also talk about containment. Not the cinematic kind with sirens and steel doors, the slow kind with careful planning and lots of uncomfortable meetings. Because the drive is not just an edit. It is an edit that is designed to reproduce itself inside a population, like an idea that refuses to stay private. If you are a mosquito in this story, you do not feel like a villain. You feel like hunger with wings. You feel heat from mammal skin and the soft breath of CO2 and the simple math of blood. And then, inside your cells, there is a copied instruction that your ancestors did not have. It biases inheritance. It turns the normal coin flip of genetics into something more like a loaded die. And you keep flying anyway, because mosquitoes do not read policy papers. In the lab, people build gene drives by putting the CRISPR machinery into the genome itself. They include the gene for a Cas protein. They include the guide RNA that targets the matching wild-type sequence. So when an organism is born with one copy of the drive and one normal copy, something happens inside its germline. The Cas cuts the normal copy. The cell repairs the cut by copying the drive sequence as the template. And now, instead of one drive copy, there are two. Congratulations, the drive has converted a heterozygote into a homozygote. Then, that organism has offspring, and most of those offspring inherit the drive. Not half, more than half. Enough to let the drive spread through the population, generation by generation, like a rumor that keeps getting repeated. This is the part that sounds clean in theory, but theory is a quiet room. Ecology is not. In the real world, there are complications that are as old as life. Resistance can evolve. Sometimes the cut site mutates so the guide RNA no longer matches well. Sometimes the repair process introduces changes that block future cutting. You end up with alleles that are drive resistant, and those alleles can spread too, especially if the drive imposes a fitness cost, because a gene drive is not free. It can reduce fertility, or survival, or overall reproductive success, and natural selection notices costs with the enthusiasm of a debt collector. So gene drive research is full of careful modeling. People simulate population sizes, mating patterns, migration, seasonal changes, and mutation rates. They ask whether the drive would spread, stall, or collapse. They ask how quickly resistance might appear. They ask whether a drive could be localized, limited to one region, rather than taking a global tour. This is where you discover something almost comforting. Even with powerful tools, biology is still hard to control. The world is messy enough to resist your neat plans. So, researchers propose designs meant to be more controllable, Some drives are meant to be self-limiting. They spread for a few generations, then fade. Some are designed with thresholds, where the drive only spreads if enough individuals carry it. Some ideas involve reversal drives, meant to overwrite a previous drive, which is a strange thought, editing the editor and then hoping the second solution behaves better than the first. If you are the mosquito again, none of this feels like destiny. It feels like ordinary life under ordinary evolutionary pressure. You hatch in water, you feed, you mate, your genes replicate. And whatever humans did in their bright rooms becomes part of your quiet inheritance. That is why the ethics around gene drives is a different kind of ethics. Not only individual consent, also collective risk, community engagement, international coordination. Because the unit being edited is not one patient, it is a population. And the side effects are not limited to one body. They can become geography. Now, the story can drift away from gene drives because they are only one edge of the CRISPR world, and the edge is always noisier than the center. Back in the calmer microbial universe, most of what is happening is still bacteria and phages arguing in genetic fragments. And there are more gentle wonders there. If you let your attention shrink back down, because bacteria do not just defend themselves, they also share. In the microbial world, gene sharing is not scandalous. It is Tuesday. Horizontal gene transfer is the reason bacteria can adapt so quickly to new threats. A gene for antibiotic resistance can appear in one species, then show up in another. Not because they are related like siblings, because they traded. There are a few main ways bacteria move genes around. One is transformation. That is when bacteria pick up naked DNA from the environment. A dead cell releases genetic fragments, and a living cell takes them in. It is scavenging, but for information. Another is transduction. That is when a phage accidentally packages bacterial DNA and delivers it into another bacterium. It is a delivery mistake that becomes an evolutionary event. And then there is conjugation, the one that sounds vaguely inappropriate, because it is. Two bacteria connect through a pilus, a thin protein tube. A plasmid copies itself and moves across. It is not romance. It is data transfer. In a crowded and poorly regulated network, plasmids are small circular DNA molecules that can carry all sorts of useful traits: resistance genes, metabolic enzymes, virulence factors. They are the flash drives of the microbial world, except nobody asks before plugging them in. Now you can see why a system like CRISPR matters beyond phage defense. If foreign DNA is constantly arriving, you need policy. You need a way to decide what to keep. Some bacteria use CRISPR to target plasmids, preventing conjugation from changing their genetic identity. That can be protective. It can also be limiting, because sometimes plasmids bring the genes that let you survive an antibiotic. So in some environments, having active CRISPR is a disadvantage. It blocks helpful imports. In others, it is a lifesaver. It blocks parasites and lethal phages. This is why, across bacteria, you see a patchy distribution of CRISPR systems. Some lineages keep them, some lose them, some carry broken remnants, like an old lock rusting on a door that no longer closes. There is also another quiet reality. CRISPR is not always just defense. Sometimes it looks like regulation. There are bacteria where CRISPR targets the bacterium's own genes, or near enough to matter. In those cases, the system can influence gene expression. It can shape behavior, metabolism, or virulence. It is still guide RNA and matching, still binding and interference, but the target is not an invader. It is the self-adjusted, like a thermostat, and if that makes you uneasy, good. It should, because it reminds you that biological tools are rarely single-purpose. Life reuses parts. It repurposes. A defense system can become a control system. A weapon can become a sensor. A mistake can become a pathway. Now, since we are drifting through microbial defenses, it is worth lingering on those older systems for a moment. Restriction enzymes are the classics. They were discovered before CRISPR was understood. They became the first widely used genetic scissors in molecular biology. They cut DNA at specific recognition sequences, often palindromic sequences, meaning the sequence reads the same in opposite directions, in a certain paired way. It is the kind of symmetry chemistry likes. A restriction modification system usually includes two parts: a restriction enzyme that cuts foreign DNA and a methyltransferase that tags the host DNA, methylating specific bases. The host DNA gets a chemical stamp. Foreign DNA lacks the stamp and gets cut. It is simple and ruthless, but it is not adaptive like CRISPR. It does not learn new targets. It is born with a set of sequences it recognizes, and it enforces them. And humans took those enzymes and used them for decades. They built cloning strategies around them. They made maps of plasmids using cut patterns. They stitched genes together with ligases. Before CRISPR became famous, restriction enzymes were already the quiet backbone of genetic engineering, which is fitting because they also came from bacterial paranoia. Now, bacteria also have defenses that feel almost like booby traps. Toxin-antitoxin systems are 1. 2 genes, often paired. One produces a toxin, something that can shut down translation or replication. The other produces an antitoxin, something that neutralizes the toxin. As long as the cell is healthy, both are made. Balance holds. But if a phage infection disrupts gene expression, the antitoxin may degrade faster than the toxin. Then the toxin wins. The cell shuts down. The phage loses its factory. The individual bacterium dies, but the local population may benefit. It is not noble. It is statistical. And there are still more systems, more acronyms, more defense islands. Bacteria are small, but they are not naive. They have had billions of years to practice being attacked, so they are layered with contingency plans, which brings us back gently to the human discovery story again. Because one of the strangest things about CRISPR is how long it sat there, visible, before its meaning was understood. People saw the repeats, they cataloged them, they named them, and for years they did not know they were looking at immune memory. That delay is not stupidity, it is normal. Because biology does not announce its function, it hides it behind repetition and noise and context. And context is expensive to learn. You have to notice the spacers match phages. You have to notice new spacers appear after infection. You have to link sequence to phenotype, meaning resistance, and even then, you have to identify the machinery. Cas proteins, processing steps, targeting requirements, the little adjacent motif that prevents self-destruction. Each piece arrives slowly, like a puzzle assembled in dim light. In that sense, CRISPR is not just a tool story. It is a story about how humans learn, with patience, with database searches that finally work because someone else sequenced the right virus, with experiments that feel boring until they line up, with industrial problems like phage infections ruining dairy cultures, pushing people to pay attention, and with the simple act of looking at something weird and refusing to stop looking. Now, let your mind settle back into the microbial world one more time, because bacteria do not only exist as solitary swimmers, they form communities, biofilms, the slimy layers that grow on rocks, pipes, teeth, and catheters. A biofilm is a city made of cells and glue. The glue is extracellular polymeric substance, a mix of polysaccharides, proteins, and DNA. It holds cells together. It traps nutrients. It slows diffusion. It makes life harder for antibiotics and disinfectants. If you have ever tried to scrub dried residue from a dish, you understand the general mood. Inside a biofilm, bacteria communicate chemically. They release small molecules. They sense density. They change gene expression depending on how crowded they are. This is quorum sensing, which is a polite name for the fact that microbes coordinate when it benefits them. In a biofilm, phages still exist, but movement is different. Access is different. Defense is different. Some phages can penetrate. Some get stuck. Some evolve enzymes that degrade the biofilm matrix, and bacteria respond in turn. So CRISPR in the wild is part of a larger ecosystem of protection and exposure. It is not the only story. It is one chapter in a book written in slime. Now, there is another gentle wonder in microbial life that connects back to CRISPR in an unexpected way. Bacteria can carry something like immune memory even without CRISPR. They can alter receptors on their surfaces, preventing phage attachment. They can phase vary, flipping expression states on and off, so a population becomes heterogeneous. Some cells express one receptor, some express another. A phage that binds one type cannot bind the other. So even if many cells die, some survive. It is like having a neighborhood where not everyone uses the same lock. This population-level diversity is another kind of defense. Not memory in one genome, but variability across genomes. And it reminds you that survival does not always mean one strong shield. Sometimes it means being unpredictable. Humans, of course, took notes here too, because unpredictability is also a tool. In labs, people now use bacterial CRISPR arrays as recording devices, not only to store viral spacers, but to store information about events. This is not science fiction anymore. Researchers have engineered systems where bacteria acquire spacers in response to specific signals. So the CRISPR array becomes a timeline of exposure, a biological tape recorder, written into DNA. It is still early, still limited, but the concept is quietly astonishing. A bacterium writing a log file into itself, not because it wants to, because you engineered it to. Congratulations, you have turned microbial paranoia into data storage. Now, before that feels too clever, remember the reality. A bacterium is tiny. It mutates, it divides, it loses sequences, it shuffles. So the record is noisy. It is not a perfect archive, it is a sketch. But even a sketch can be useful if you understand the noise. This is a theme in biology. You rarely get a clear signal. You get a cloudy one, and you learn how to interpret clouds. Now, speaking of signals, return briefly to those diagnostic uses of CRISPR, because that is one of the more soothing applications, in a way. Not editing bodies, but detecting molecules. Systems like those based on Cas13 or Cas12 can be used to detect viral RNA or DNA. The trick is collateral cleavage. A guide RNA brings the Cas enzyme to a target. When the target is present, the Cas activates. Then it begins cutting nearby reporter molecules. The reporter is designed so that when cut, it produces fluorescence or a color change on a strip. So detection becomes visible. This is useful because it can be fast, it can be specific, and it can be done without massive sequencing machines. It is an example of taking a bacterial emergency response and turning it into a quiet measuring device. A phage defense system becomes a clinical test. That is the strange poetry of applied science. Now, in the calmer corners again, there is something almost intimate about how CRISPR works. It is all about matching a guide RNA pairing with a target sequence. Base pairing. The simple rules: A with T in DNA, A with U in RNA, C with G. Tiny hydrogen bonds, weak individually, strong in groups. The same principle that lets your cells copy their genomes faithfully also lets bacteria identify invaders. And it is the same principle humans exploit when they design guides. You choose a sequence, you check it against the genome, you avoid close matches, you account for the adjacent motif requirement, you synthesize the guide. Then you let chemistry do what chemistry does. Bind what fits, cut what is marked. It is almost boring in its elegance, and that is why it spreads so widely. You do not need to understand everything to use the core. You need to know how to aim. But aiming is a science too. Guides can fail. Chromatin can block access in eukaryotic cells because DNA is wrapped around histones and packed. The target might be hidden. The Cas might bind less efficiently depending on local sequence features. So researchers developed scoring algorithms predicting guide efficiency. They test multiple guides per gene. They build redundancy into screens. They accept that biology is not a vending machine. And when they need more precision, they engineer the tools further. High-fidelity Cas variants designed to reduce off-target cutting. Cas nicases cutting only one strand, used in base editing and prime editing. Split Cas systems, assembled only under specific conditions. Inducible expression, turning the system on only when needed. Again, boring engineering, the kind that prevents expensive regret. Now, as the story keeps drifting, you can feel the scale shift again, from human ambitions back to microbial routine. Because while humans debate what to edit, bacteria keep doing what they have always done. They acquire spacers, They lose spacers, they survive phages, or they do not. They share plasmids, or they block them. They form biofilms. They compete. They cooperate. They persist. And it is worth remembering that in many environments, CRISPR is not even the dominant defense. Some bacteria rely more on restriction systems. Some rely on surface variation. Some rely on abortive infection. And some do all of the above, layered like locks on a door in a bad neighborhood. The microbial world has no universal strategy, only whatever works long enough to replicate. Now, there is one more quiet thread that connects back to the original discovery, and it is about how humans recognized meaning in the spacers. Because matching a spacer to a phage genome is not always straightforward. Viruses mutate, databases are incomplete, spacers are short. So early on, many spacers had no known match. They were called orphan spacers sometimes, unknown origins. They might come from phages that have never been sequenced, or from plasmids, or from mobile genetic elements that exist only in certain niches. This means CRISPR arrays can also be used as ecological probes. If you sequence bacterial CRISPR arrays from an environment, you get hints about what viruses are there. Even if you never isolate the viruses, it is like reconstructing a predator population from scars on prey. Not perfect, but informative. Researchers have used this idea in oceans, soils, and human microbiomes. The gut is full of bacteria. The gut is also full of phages, a whole virome living quietly alongside the microbiome. And those phages can shape bacterial composition, which shapes digestion, immune interactions, even drug metabolism. So when you look at CRISPR arrays in gut bacteria, you might be seeing a history of phage pressure inside a human body. Not your immune memory, but theirs. Different layers of defense, nested. It is hard not to find that slightly unsettling. You are not alone in your own biology. You are an ecosystem with tenants who have their own enemies. Now, hold that thought, because it leads naturally to another gentle point. CRISPR is often presented as a clean editing system, but in nature, it is also a force shaping evolution. If a bacterium targets a phage sequence, phages that mutate away survive. If phages carry genes that help bacteria, bacteria might tolerate them. Some prophages integrate into genomes and become permanent features. They can carry toxins, secretion systems, metabolic genes. Sometimes bacteria become more dangerous because they carry prophage genes. Cholera toxin, for example, is encoded by a phage. So phage biology and bacterial biology are tangled. CRISPR influences that tangle by limiting which phages can persist, which genes can move, which mobile elements succeed. So, when humans discovered CRISPR, they did not just discover a tool, they discovered one of the hidden regulators of microbial evolution, a system that shapes who gets to exchange genes and who gets cut off, a border policy written in nucleotides. Now, speaking of borders, there is another odd twist. Some bacteria and archaea have CRISPR systems that target RNA. That changes the logic. Instead of destroying viral DNA, they destroy viral messages. It is like silencing a broadcast rather than breaking the transmitter. Cas13 is the famous example. Human